Hi there, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about the DNA replication termination in eukaryotes. Previous my videos, I already talked about the initiation and the elongation steps. So always remember that the DNA replication in eukaryotes and prokaryotes are different. So I'll make another video for the prokaryote DNA replication. So first, I would like to say that after copying most of the replication, when the replication fork come close together, then the termination process start. So let me draw a picture. So this is the replication fork. So here the CMG complex and all of the replication associated proteins, that is a replisome, and here in this strand also there are some this complex. So when they come closer, then the termination process is started. So let's have a look the diagram that I have. So first, in this figure, as you see that in the in between there are some part and some DNA are non-replicated. So when most of the DNA is replicated and there are some stretch of the DNA is not replicated, and in that time the CMG associated proteins and complex, they come closer and allow the formation of the supercoils in unreplicated DNA. All right. So then convergence occurs. So in the next step, that is a convergence, and the bypass occurs, that both of the complex, they bypass each other. And next step is after bypass, the CMG helices keep translocating. As you see that the bypass already is taken place, and then this complex it keep translocating until they pass over the single stranded DNA and double stranded DNA junction of the downstream Okazaki fragment. Alright, and then the final Okazaki is processed. So in this process, as you see there, the polymerase delta, it processed the final Okazaki. And there, as you know, that in the lagging strand there is the overhang region in the 3' region. So that 3' region overhang is processed by the FEN1 enzyme. And in the next step, that is the final step, and in this step the CMG encircles the double-stranded DNA. So as you know, in the termination process, that the double-stranded in the termination process, in the last step, the double-stranded DNA, it get encircled. So as you see in this figure, that double-stranded DNA, it get encircled, and once this encircle looks like this kind of figure. So this is the encircled, and then once double-stranded DNA, it get encircled, then CMG undergoes polyubiquitination on its MCF7 subunit by SCF. SCF, this is the enzyme for the ubiquitination, and this E3 ubiquitin ligase, that is the SCF complex, and that SCF complex contain 3 different protein: Qlin, SKP, and F-box. And then in the MCF7 complex, in the lysine residue K48, lysine 48 residue, the ubiquitin protein, it attached. So in this here, so I would like to mention that what is ubiquitin actually. So ubiquitin actually, this is one of the step for protein degradation. So as you know that the proteasomal degradation, so as the same kind of degradation is the ubiquitin. So when the ubiquitin protein, it attach with any junk protein, then it get degraded. So then the next step is the MCM7 polyubiquitination and CMG dissociation both require the E3 ubiquitin ligase SCF. So for both this, it required this ubiquitin. And the next is, this is the recently found that CRL2LRR1, this is the choline ring ligase 2 associated with LRR1, that is one of the receptor proteins that promotes MCM7 ubiquitination. So this is a recently found. And next step, when the CMG is get polyubiquitinated, then the ATPase 97 P97, that is known as also VCP, that proteins, it removed the CMG from the chromatin, from the chromatin. So then the final step of the termination, it's occurred, and then it get terminated. So this is all about the DNA replication termination process in eukaryotes. So if you like this video, kindly hit the like button, share it, and please subscribe my channel. Thanks.